Hey everyone, this is Stephanie, and I am going to be doing a review for our upcoming lab quiz that is tomorrow. If someone didn't have a chance to take a look at this, I decided to go ahead and make this little review, so I hope this helps. In this review, I'll be covering pretty much everything that went through in the study guide. 1, 2, 3, 4, 5, 6, 7, 8, 9. I got 9 pages of review for you guys from the entire lab, and I went into detail so that to rather give you more information than give you less would make me really happy, so that we can make sure that we cover all of our bases. I also have on deck the sheets that was given out from our professor that might help everybody for a quick review. And in the lab review that she gave us, she has gram stain, acid fast, endospore stains as being like pretty much what the gram stain, acid fast stain, and endospore stain being the most important stains that we need to understand. And in each of the categories, we have the primary stain, the mordant, the decolorizer, the counter stain, and then you have kind of a follow-up question at the end. On the other page, we have a drawing of basically please draw and define the growth for each of the FTMs. So that's our lab that's talking about aerobic and anaerobic life. And then, organisms. And then we also have motility. And then we have microscopy. And so I'm going to start and go into detail with these, essentially what everything that needs to be covered. And so again, this is going to be in really deep detail, starting from safety going all the way to motility, since that was the last thing covered. So let me just charge really quick my laptop here, my iPad, and we'll get right into it. Also, a note, anything that you see in purple is going to be questioned. I really think that she's going to focus on. I've noticed our professor has a way about like ignoring the things that are obvious and that she's emphasized on, and rather she'll explain it thoroughly, but she might not emphasize on it because she really wants you to understand it, and if she's mentioned it or she's talked about it, she wants you to make sure that you completely understand it. So I'm going to explain it to you, and your challenge is to explain it to somebody else in the class, or even if you don't have anyone that you talk to in class, and explain it to your cat or explain it to in some format to show that you actually understand it. Okay, starting from week 1, I'm going to have my pen here. All right, so starting from week 1, this is what we covered in week 1. We did lab, quote unquote, week 1. This is week 1 and 2 smushed together. We did lab guidelines, safety. We did microorganisms in the environment. We did morphology, aseptic technique, and streak plate for isolation. So that's what we covered in pretty much week 1/2. So I'm going to be covering those really quick. And then anytime that you see purple or a star, purple stars, that essentially means I'm pretty sure she's going to ask us a question on that, because she can only ask us so many questions. I'll try to go through and highlight as many questions I think she's going to ask. Okay. Really quickly, in terms of safety, I'll start here. Safety. If personal items get contaminated, they have to be autoclaved. So this was actually in the safety guideline. And so just in case you have like your own personal item, something happened to it, it will get autoclaved. So if she asks that, boom, there's the answer. It will be autoclaved. Here's another question. Upon leaving, you have to spray with disinfectant for 10 minutes. So this is just when you come into the lab, when you arrive and when you leave, boom, you gotta spray. And you have to leave it on there for 10 minutes, and then you wipe it off. So that was another question. In case of a spill, warn the professor. Soak it for 15 minutes if there's a spill with a towel on top, and notify the professor. I don't know if that'll be a question, but that was in the guidelines. So these are all pretty much simple things. If you're watching this and you're not in our class, these are the ways that our guidelines are in our class in San Diego. But if you guys have a different format, then you want to go with that format. Okay, BSLs, biosafety levels. Here we are. So biosafety levels. So we have different levels in the ways in which we handle organisms. And so essentially we have 4 of them that we utilize in the microbiology lab. Number 1 is BSL 1. Okay, this is minimal threat. And this can be open-handled. And the organisms that we talk about when it comes to BSL 1 is E. coli and B. subtilis. There's another one, and there's actually many other ones. But I think the main point here is that these biosafety levels have minimum threat, but you still have to have the standard safety handling them, right? With gloves, goggles, all that to protect yourself. All right? She's not going to ask about BSL 1 because BSL 1 is too easy. She's not going to ask about that. I think she's going to ask about either BSL 2 or BSL 3. So when it comes to BSL 2, it's a moderate hazard, okay? This is bacteria that's encountered in the community or in the environment. They may cause human diseases, but they're pretty much treatable. So we have Salmonella, C. diff, and Staph aureus. So those are the organisms that are associated with this bio safety level. And so that's what you want to focus on. I think the main term is moderate hazard encountered in the community. Then you have BSL 3, which is serious/lethal. And these are local and exotic encountered. Locally and exotically encountered. And so the treatment we may or may not have a treatment for this. So, for example, with tuberculosis, we do have a treatment for tuberculosis. It's a very long treatment. But we do have a treatment for it. And then you have B. anthracis, okay? That's associated with anthrax, you know? So you realize that this can be a lethal organism. So that's going to be huge. So for BSL 3 and BSL 2, I gave it purple stars because I think that these might be on the test. I'm not certain, but that would be my guess. BSL 4, she's not going to put this on there, right? Like, this might be on there, but I highly doubt that I think she realizes that we know that Ebola and Marburg and Lassa, like Ebola is going to be associated with the BSL 4 because it's very highly potential lethal. So she knows that. We know that. So I think she's going to give us one that's a little bit more tricky because it is an exam after all. Little tiny note. Autoclave. Autoclave occurs with steam at 121 degrees Celsius. And we have to remember that I don't think she's going to ask this on here because this is our lab exam. This was already covered. But the fact that these do not inactivate prions. Okay? So we have when we do autoclave, it's going to occur at 121 degrees with steam. So I don't know if that's going to be on there, but that's something important that she mentioned a couple times. So there you go. All right, moving on to 2-1. Woo! We made it through the first part. We're going to keep going, and I'm going to try and be as thorough as possible and highlight the important things that might come up. Now, let's just move right through it. All right, background. Microbes are ubiquitous in nature, right? We find them in the soil, air, and water. And here's a terminology that I'm going to highlight here. It's pathogenic, which means disease causing, right? Or infectious. All right, so when something's pathogenic, it's disease causing. Definitely a question that will be asked, if it's my guess. You can already assume that a non-pathogenic organism is benign, right? Because we said pathogenic is disease causing, therefore non-pathogenic is benign. And then these are mostly single-celled organisms. So then when we get to actual terminology, we have culture media. Culture media essentially is for growing organisms, okay? We have the FTB later down the line that we utilize for growing organisms. And so culture media is utilized for growing organisms. So they would grow and multiply. That's my multiplication right there of an organism. That's drawn horribly, but hey, I tried. What does it supply? So here's a question that she might ask. So what does culture media supply? And your response would be carbon or nitrogen, right? It'd be tricky here, right? Because in the form of right here, it can be in the form of carbs, right, which are mono or polysaccharides, peptides, amino acids, nucleic acids, lipids. But all of these in this form supply carbon and nitrogen. Okay? So then what does it supply? It supplies, so the culture media supplies carbon and nitrogen in the form of carbs, peptides, amino acids, nucleic acids, lipids. Okay? So bringing that all together. So then you have your nutrient media, right? So your nutrient media can be in two forms. And one form is liquid and the other form is a solid. Our agar is the solidifying agent, right? And then we have our broth, which is the liquid form. All right? So now let's get into another term. When you talk about, talk about inoculation, all right? All inoculation simply is the transfer of microbial cells to a nutrient media, right? And when we do that, we tend to do that aseptically, which means without contamination. I have this term later down the line, so you will see this again. And so you might ask, okay, so once we transfer, how is growth observed? You might ask. So it's observed in two ways, right? Because I told you we have the broth and the agar, which is the liquid and the solid. So the way it's actually viewed is via if you're looking at, for example, the liquid, we're going to look for turbidity. We know that turbidity means cloudiness. Is she going to utilize this? No, because we all know what turbidity is now. Turbidity just basically means it's cloudy, or the tube would be cloudy. It shows cloudiness. And when it comes to solid, the agar plate, we look for discrete masses or large mass of cells. That is going to be our affirmation of growth. Okay, so boom. That's all it is for 2-1 for microorganisms. I'm going to keep moving on, and feel free to email me if you have any questions about this. But I'm just covering the bases here of what I highly think going to cover. Okay. And try and throw some tricks at you. Okay. Now we're in cell morphology. Now this is really important, right? Because this is when we actually grow the bacteria in a nutrient, whether it's an agar plate or nutrient media. Let's talk about morphology. So the thing that I think is messing many people up, including myself when I was creating this, was the fact that plural, which means many, and singular, which means just one, obviously, can get confusing because when they utilize nomenclature to describe different bacteria, when they use nomenclature is like the terms, and you're talking about many, plural, or few, single. When you talk about that, we utilize the Latin form because this is used all around the world. And this is used, you can talk about this in Germany, in France, in Japan, in Africa. You could talk about this in many, different countries and continents. And when you talk about that, it pretty much would be the same thing, because scientists have to be able to talk about this in one language when they're describing a species. So because the Latin, the Latin language got to it first, this is how it's named. So when you talk about singular, singular, and plural, like I said, singular is one, plural is an example here, right? So if you're in the sea and you happen to see an octopus, you see one octopus, 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 one single octopus, all right? But then, say for whatever reason you decided you kind of invaded their privacy and you run into a whole group of you wouldn't say octopus. I don't want to say what. I don't want to add an S to that because that would be incorrect. The correct way, or the plural form of octopus, is octopi. So you would a family of octopi, or if you just saw one, it'd be octopus. The octopus is a singular form of one animal. All right, right. In the same way, when it comes to bacteria, the same way when we talk about morphology and arrangement, we're talking about the shapes, and we're talking about if they are singular or plural, we have the same thing. You see how octopus, octopus, right here, kind of correlates with this. We have a sphere, okay? You're looking in the microscope and you see one sphere, then you have coccyx. Coccyx. And I hope I'm saying this right. And then when you have many, plural, and then you have many spheres, then you have cocci. And so when you look at rod or rod, if you have one singular rod, it's bacillus, which is single, one single bacillus. When you look in the microscope and you see many of them, many rods, you say bacilli. Just like octopi, you see a whole family of octopi, you'd see bacilli. You see bacilli, multiple. And that's the plural form. So if we keep down this path and we start looking at the other forms of bacilli, which is many rods, you also have curved rods, which are called vibrio. And then you have short rods, which are called co-bacilli. And then short, fat rods. So if you see a short, fat rod, that's your co-bacilli. The reason why I drew it in brown, because it reminds me of a coconut. They're short, they're fat, they're round, except for it'd be a spread out one that's kind of short and longer. But co-bacilli is short, fat rod. It's not quite circular. And so that's the name. And they're all seen in pictures. And we'll get down the arrangement down here. Okay, so then let's now talk about a spiral. If you see a spiral, okay, if you see a spiral, then you have more than one spiral. You have either spirilla, which is the single form, but you have spirals, many spirals. So this one spiral, it's called a spirilla. If you have multiple spirals, that's my best attempt at a spiral. Multiple spirals, then you have spirillum. Spirillum, that's me. Think of when you see the M, think of many when it comes to this. Spirillum, many, and spirilla, and that's just singular form, all right? So here's my singular form, and this is my plural form. When it comes to flexible spirals, all right, you have spirochete. so that's our last example. Okay, so this is my spirochete. Sorry, flexible spirochete. Spirochete. All right. So here's a hint when it comes to arrangement. All right, a spirilla are single celled, and when we talk about arrangement, and just remember this for arrangement. You have arrangement is essentially the number of plane divisions and if they separate. Okay, so when you talk about these plane divisions, all right, we have single cells, we have pairs of cells, chains, clusters. All right, so we're talking about the plane divisions, right? And so when it comes to a single cell, you can see a single cell that's a spirilla. I don't know if I'm even spelling this right. Let me double check. Spirilla. Yeah. Let me add an L here really quick because I spelled it wrong. So sorry about that. Okay. So your single cells are going to exist in cocci, and I did this to try and help you remember. Single cells exist in cocci, bacilli. And when I put this cocci, I'm talking about you see many single cells on their own, or you see bacilli, which is many rods on their own individually, right? These are single cells individually. You have spirilla, which is single cells, and then you see spirochetes. Say you might have spirochetes. It means that you're seeing many flexible individual rods on their own. And then you have vibrios. When it comes to pairs of cells, okay, now you have this term called diplo, right? So you may have a diplococci or diplobacilli. All right, down here I have a diplococcus, and then I have two of them. So now I have diplococci because there's two of them. Here's one, here's two. Does that make sense? This is one diplococcus, and then this is two of them. So diplococci, I have many of them, or multiples of them, two of them. And so, but it's a diplococci because it comes in pairs of cells. And then you can see this also exists down here when you see this diplobacilli. So yes, you can have a diplobacilli, right? You have a pair of bacilli, a pair of rods connected. Then you have chains, okay? And these chains exist, so you can have, in terms of when it comes to a chain, you can have streptococci or streptobacilli. Now I'm just gonna try and throw a trick at you. Are you going to be able to see like streptospirilla? If you said yes, then you'd be wrong, because as you can see here, it shows chains. You can only have a streptococci or streptobacilli. You can't have streptospirilla. You can't have chains of spirilla, of spirals. They exist as single-celled organisms, and they will not be connected as chains. And then lastly, we have clusters, right? Staphylo, okay? So same thing here in terms of giving you a trick. You have staphylo, which is basically cluster. It's a cluster right here. And they will appear in grape-like. And you see the only morphology here I have is the cocci. So you're not going to see bacilli, staphylobacilli. That is not something that you're going to see. You might see bunching or, cells close together, but you're not going to see, there's no thing that we have or terminology as staphylobacilli. We have staphylococci, and that is the only one associated with clusters or grape-like clusters, okay? So that's why I made this little trick here. You're not going to see Streptospirilla. It's just not going to happen for our purposes in class anyway. Okay, so that's cellular morphology and arrangement. I hope that octopus and octopi and stimulus and stimuli was helpful for you to not confuse them, because it is easy to get confused. Moving on to colony morphology. Now we're talking about colony morphology. Remember, when we talk about colonies, we're usually associating that with a plate. Boom. That's my plate right there. And let's go through some terminology. Oh, let me go back here. Let me go back here for a second. I'm going to hazard a guess that she's going to harp on Vibrio as a question. Flexible spirals, which are spirochetes. Don't forget that. Spiral, singular, spirilla, or spirillum. I could see her asking questions on that. In terms of this, maybe a question on bacillus, single, and that would be my guess for what she would probably ask. Okay, so I hope that helps. Let's go back to 2.1. So we're moving forward now. We're going to go talk about colony morphology, and I've got some terminology that I'd like to share. We have mixed culture. And all a mixed culture is that you have a multitude of species. So instead of one pure species, you have a multitude of species. And here's another term, isolated. This basically means that the colonies are not in contact. They're not touching. So for example, if you do a plate, here's my example. And I'll use the one that we often do, our professor loves to give us, is the quadrant streak. When we do the quad streak, the point of doing that is to isolate bacteria. So say I have, some nutrient culture and I aseptically inoculate this plate. So I'm going to do this, right? That's my first streak. Then I turn, I go through the aseptic technique of transferring. I sterilize my loop, and then I come on this side. And I drag sorry, I'm going to just do this in another color so that I've flamed my loop, and I'm dragging from here, and I'm trying to isolate these colonies. Now I come here, and then lastly I do this. And I usually end up with 1 or 2 colonies all isolated, all by its lonesome. That's the goal anyway. How often does that happen? We do our best, right? So these right here then would be the isolated colonies that aren't in contact. You see how these all are contact in each quadrant, but the last quadrant is not in contact. That is when you isolate the species. So then when you do isolate the species, the pure culture, you can make a pure culture, and a pure culture is one species. So now I'm essentially assuming that I have one species from that isolation. And this colony is a visible mass of cells, right? So this is like after this is incubated, we incubate this for however many hours our professor expresses us to do, because we don't know too much about incubation right now. Incubation. Now we go ahead and incubate it, and then we we have this visible mass of cells, and we isolate it. And so this, like I already said, this is our one species, and we're trying to separate that out from the visible large mass of cells that we've created when we did our initial plating. So I'm going to go into the theory that our professor asked us to talk about, and I'm just going to talk about really quick questions that I think she might ask. For example, so in the theory, it says, okay, genetic makeup. So what is the genetic makeup? So the genetic makeup of a species is the color, the size, the shape, the texture. Those are your main things that genetics have a role in. But when it comes to influence by, right? So she can ask the question, okay, question. What are colonies influenced by? Name 3 things. She could say nutrients or nutrient availability, time, which is the incubation period, right? If you have more time, you have a larger chance for growth. Temperature, there are certain temperatures that a species will turn a certain color, right? If you put one in 30 and you put one in 37, at the cooler temperature, one might turn red, right? So that has to do with what it's influenced by. But that doesn't have anything to do with that doesn't have to do with its genetics, its genetic makeup. That has to do with how the species is, what the species is influenced by. Okay, so, and then here's this portion right here. It's pretty inundated. I think this is going to be quick to run through, though, and I think it's going to be important to know, like, at least 1 or 2 of them, just in case she asks a question. So then, we have shape, or size, margin, shape, surface, texture, elevations, and couleur. So if I were to hazard a guess, she would probably ask us this. So she'd probably ask us to define what something that's punctiform. Okay? Basically, something that's punctiform is it's tiny and pinpoint. Right? So that's just a guess. I don't know if she would really ask that, but trying to get us to explain, for example, what a shape is, what it looks like, not just, having all these words out, but making sure we understand what it actually looks like, so when we're identifying, we can explain them properly. She might use undulate, which just means wavy, rhizoid, which just means like branch-like, filamentous, which means hair-like. Filamentous. That's me trying to draw hair, you guys. Please don't judge. I'm just trying to look for the terms that seem the most like we could possibly mess these up. But I don't think we'll mess these up. Butyrus, which basically means buttery looking. Essentially looks like melted butter. I don't think that we'll get any samples that look butyrus, but if we do, we have an adjective to describe it, so good for us. And that's about it. Let's see. This one, bonnet. Hopefully that's visualized. Bonnet means it looks button-like. So this is talking about the elevation. That means the center would look kind of raised. Yeah. And the last one's in terms of describing our morphology is going to be opaque and translucent, obviously. Opaque means is you can't see through it. That's all it means. So if she asks you to just, what does opaque mean, which I think she will, but if she does, it means opaque. You can't see through it. Whereas translucent is the opposite. Cool. Hope that helps. Moving on to aseptic technique. You guys, check out my beautiful Bunsen burner. I worked really hard to draw this for you guys, so I hope this is helpful. Okay, so we only needed to understand the theory only. So when it comes to this, I'll just try and cover some of the main things I think that she's gonna. Culture. So basically, a culture is a medium that has living microbes. We have pure culture, which I already explained, that it is a single species. And when you aseptically transfer something, you do this without contamination. And you can get contamination from anywhere. You can get it from the bench, from the environment, from your inoculum. You could accidentally touch something. You can get it from almost anywhere. And so aseptic technique just basically means you're trying to not contaminate your sample. Excuse me. It's like, I don't know if you know this, but it's like 11:27, so I'm exhausted, but I really want to share this with you guys. Okay, so a word about the Bunsen flame. Okay, so here's a quick word about this. If she asks you a question time, you guys know how much I love questions. Question time. So she might ask you a question, something to the effect of, okay, sterilization of inoculating instruments is conducted how? That's the question. So if you're sterilizing your gorgeous inoculation loop, that's my best attempt at an inoculation loop. Okay, I'll try harder. Okay, that's my inoculation loop. Thank you. Thank you very much. Okay, so when it comes to the inoculation loop and you're sterilizing it, how do you do so? Okay, so here's the answer. The answer is right here. It's the tip of the inner cone, right? Here is my outer cone right here. This is my outer cone. And my inner cone is right here. This is my inner cone right here. So then the correct way in which to sterilize your inoculation loop would be the tip of the inner cone of the flame, right? Whereas, how about if you're going to do heat fixing? If you're going to do heat fixing, or sterilize the mouth of, for example, your bacteria, that's your nutrient or whatever, and you want to sterilize the mouth of it, what you have to do is you have to utilize the outer cone, right? So there's a huge difference. I don't know if she's going to ask this, but if I were to hazard a guess, I could totally see her, wanting to make sure we know precisely where and to inoculate. So there's that. Okay. So that's covered. Boom! On to streak plating. I hope you enjoy this. This was easy. I just had to because it's streak plating. Do the quad streaking, right? And the whole point of doing this is to try and obtain the original cell type, just the one species. Because if you have more than one, then it's going to be really difficult to, identify what kind of organism you have. And when it comes to a medical implication, that's going to be huge. Okay, moving on. So a CFU. So this was really confusing for me, but I think the easiest way to describe this or explain this is a CFU is a measure of viable cells that accounts for the fact that they attach to one another and they grow to form a colony. So, right, say that you do this isolation technique, okay? Then you go and look at it at a microscope and you get really close, you're in oil immersion, you're going to town, and the goal is to again, to isolate to get to the original cell type. So then you may see this CFU or this colony forming unit, which is just a measurement. It's a unit of measurement, just like we have different kind of units, is to account for the fact that they may be pairs, they may be chains, they may be clusters, and the whole point is to separate the CFUs on the agar surface so that individual CFUs grow in isolated colonies. Because once you isolate, you can ensure that you just have one species. That's the whole point. So yeah, it's a unit. It's just a unit of measurement. It's no different from any other unit of measurement that we attribute so that we can quantify something, put a number to something, have an amount of something, right? That's we're in science. We're using science, and it's very important for us to quantify data so that we can try and come to conclusions about things that we learned about. So that's why that's important, and I hope that helps explain what a CFU is. So again, it's a measure of viable cells that accounts for the fact that these cells can attach to each other and they grow to form a colony. They can exist. They can even exist single. I don't know if I put that, but they can exist all by themselves. Boom. That's a single cell. That's a single coccyx. And I have this looks like a diplococcus right here. And then you have chains, clusters. They can exist in so many ways. And it's again to just separate the colony forming units on the agar surface so that individual CFUs grow in isolated colonies. Moving on. How many more pages do we have? Oh my gosh, we have so much left. All right, doesn't matter. I totally hope this is helpful. I can go over this more in detail tomorrow, but I'm going to try and get as much as I can out today because I've already been going for an hour and I want to get